Hey, welcome back to It's Lit, where I try to understand health and science right from the source, the primary literature. First try! It's lit! Now, whether you like it or not, viruses exist in absolute abundance. In fact, for every millimeter of ocean water lives millions of viruses. Now, thankfully, not all these viruses cause human diseases. What's really exciting is how we're using these viruses and engineering them in order to battle bacterial infections, and the topic of this video, fight cancer. From a materials perspective, viruses are actually quite simple. They're really only built to transmit their genome from one host to another. Now, to understand how, we need to understand something called the Thelytic Cycle. To understand the Thelytic Cycle and the fundamentals of viruses, we're going to look at the HIV virus. So like many viruses, they require an outer coat. And extending out from this outer coat are spike proteins or recognition proteins. And contained within the virus is its genetic material. It contains the blueprints in order to make the virus's outer coat, its spike proteins, and all other accessory proteins. The virus's spike proteins allow it to recognize, attach, and enter its specific cell types. Once the virus has entered the cell, the lytic cycle really begins. First, the virus's genetic material is processed and incorporated into the DNA of the cell. Much like a computer virus, the virus hijacks the cell's means of production, creating its own protein coats, its spike proteins, and replicating its own genetic material, recreating more viral particles. Now, in order to infect more cells, the newly generated viral particles need to escape the original host cell. The primary mechanism of escaping this cell is through lysis, hence the lytic cycle, where viral particles will break open the cell membrane, rendering the cell dead and releasing the new viral particles into the microenvironment. These new viral particles are able to now infect other host cells, further perpetuating the lytic cycle. Okay, so now that we understand some virus basics, it's time to talk about cancer.
It's probably the one thing I know the most about in biology, and trying to break it down into a minute is really difficult.
Oh boy. To really understand how cancer occurs and how cancer works, we need to understand the central dogma of biology. The central dogma of biology is how our cells read DNA in order to produce proteins, which are essentially the workforce of the cell. When we need to make a protein, the DNA is going to be read and translated into RNA. The cell is able to read RNA and produce strings of amino acids that constitute proteins. Depending on the order of the amino acid sequence and various post-translational modifications, the ultimate shape of the protein is formed. And that ultimate shape dictates that protein's functions. Now, one of the central themes in biology is structure dictates function. When we look at organ systems, organelles, cell-to-cell interactions, and the very components that make cells work, the protein, the RNA, and the DNA, ultimately, structure dictates function. Now, pared down, when we talk about cancer, we're essentially talking about a disease of uncontrolled cell growth. Cells that both divide uncontrollably and refuse to die when needed ultimately take up space and resources. This really destroys the organized structures that allow organ systems to work. Now, as we age and or become exposed to environmental carcinogens, mutations can occur within our DNA. To understand mutations in the context of cancer, we're just going to focus on one protein called p53. p53 is really important because it's mutated in more than 50% of cancers. p53 acts as the guardian of the genome. It helps in the activation and initiation of DNA repair mechanisms. Additionally, it stops the cell from dividing if errors are present. And if the errors are unfixable, it programs cell death or cell suicide. Just by looking at its functions, you can see that without p53, a cell can easily divide uncontrollably and evade cell death. This is definitionally a cancerous state. This happens when mutations occur to the segment of DNA that codes for the protein p53. We get an altered amino acid sequence that renders p53 non-functional. So quite amazingly, by understanding how cancer cells work, scientists are able to program the genetic material of the virus in order to carry out functions that they desire. Now, the Onyx 15 virus is an adenovirus. Onyx 15 is an engineered adenovirus. Although it's not FDA approved, Onyx 15 works in a way that I can probably explain to you in the confines of time for this video. Now, there's a reason I'm bringing up Onyx 15 as an example. In greater than 50% of cancers, we learned that p53 is not functional. The Onyx 15 virus is only able to replicate in p53 negative cells. That means if the Onyx 15 virus were to infect a cell with functional p53, oftentimes a non-cancerous cell, then it will not replicate and then it will not lyse the cell membrane. Now, when Onyx 15 infects a p53 negative cell, it does replicate. And once it replicates, it will lyse the cell, rendering the cancer cell dead and releasing new viral particles ready to infect more cancer cells. Now, a different way to kill cancer cells with viruses is through an FDA-approved virotherapy called TVEC, which essentially infects cancer cells and initiates the immune response, allowing immune cells to find, target, and kill cancer cells. Now, this video is already probably way too long, so I'm not going to get into the overlap of oncogenic virotherapy and immunotherapy, but just note that the future of oncogenic virotherapy is likely to be in conjunction with immunotherapy. That's at least what seems to be the most effective way of using viruses to fight cancer. Considering how big my current subscription count is, I've been advised to ask you to kindly subscribe if you enjoy my content. And if you have a burning question or you're interested in a specific topic, please let me know in the comments below. I will see if I like it, and if I do, I will make a video about it and I will put you in the video somehow. Thank you for watching, and please stay curious.